Hey everybody, so thank you for joining us for the Institute of Food and Health Research Bites. I'm delighted you could join us this morning and very pleased to welcome Dr. Chris Shannon. So Chris completed his PhD in biomedical sciences at the University of Nottingham, and he then did postdoctoral training in the diabetes division at UT Health San Antonio, Texas. Now, his research is focusing on understanding the metabolic basis of human disease, and he uses cell, mouse, and patient models to identify novel therapeutic approaches to combat obesity-related conditions. So quite recently, Chris has joined us in UCD, moving here in November 2021, and he's pursuing an MSCA Development Fellowship and is working with Professor Helen Roach at the UCD Conway Institute. So this work is understanding how diet and the microbiome shape the progression from non-alcoholic fatty liver disease to liver cancer. So over to you, Chris. We look forward to hearing your talk. Thank you very much.
Thank you. Let's share my screen here. Okay, so thanks very much for that introduction, and thank you as well for the opportunity to present some of my work today. I am mostly going to talk about some work that was done back on my PhD at the University of Nottingham. I think it's a project that is going to be of more interest to the group here. And yeah, as a bit of background, I am sort of interested in all things metabolism and understanding how metabolic pathways at the subcellular or cellular levels drive tissue function, and how this impacts upon whole body physiology and metabolism. And in understanding these pathways, learning about interventions that we can test to improve human disease. So an overriding theme of my work to date has focused on insulin resistance and the problem of insulin resistance in chronic disease. And obviously, typically we think of insulin resistance, we think of type 2 diabetes, and perhaps obesity-related diseases such as fatty liver disease and cardiovascular disease. But there's this increasing appreciation that insulin resistance is a key component of other chronic conditions, including many cancers, neurological decline, as well as viral infection and also aging. And some of the data I present today is going to be related to insulin resistance and aging. So really, I think I like this slide because it kind of shows that insulin resistance is kind of a core pathophysiology, whether causal or consequential, of almost every major chronic morbidity. So it's clearly worthy of study. Skeletal muscle is probably the main focus of the work I'm going to talk about today, and insulin resistance in skeletal muscle. And one of the primary mechanisms thought to be involved in driving skeletal muscle insulin resistance is this accumulation of ectopic lipids or intracellular lipids, which we call IMCL. And these are lipid droplets that accumulate and build up either just within the cellular membrane of muscle cells, so these are subsarcolemmal lipids, or between the myofibrils within a muscle cell, which we term into myofibrillar lipids. And both types of lipids are believed to interfere with insulin action in various ways. And indeed, one of the most robust predictors of whole body insulin sensitivity is the level of IMCL determined by MRI or other methods. And this is quite a reproducible finding in the literature now. So one of our goals during my PhD was to come up with interventions that might reduce intramuscular lipids and whether this improves insulin sensitivity. So one of our focuses was on this metabolite carnitine. So back to the sort of biochemistry days at college, think of carnitine as a key metabolite involved in the transport of long-chain fatty acids into the mitochondria. So it's really important for fatty acid oxidation. And quite a lot of research from the exercise physiology field actually showed that the availability of free carnitine in muscle is limiting to maximal fat oxidation. So one of the goals of the group in Nottingham before I started my PhD was really to understand, can we increase muscle carnitine? And if so, can we increase fat oxidation? And muscle carnitine is notoriously difficult to increase. Although as a supplement, it's been on the market for decades, it's not very effective for numerous reasons. One is that it's got a very low bioavailability. So it's difficult to actually get into the system. Most of it is excreted in the feces or in the urine. And also the muscle carnitine transport is actually saturated at normal carnitine concentrations. So if you take a carnitine supplement, a little bit gets into the system and very little of that gets into the muscle. And so work at Nottingham discovered that when you increase plasma insulin concentrations, you can actually drive carnitine into the muscle. And leveraging this discovery, they showed that by feeding carnitine in a carbohydrate beverage, you can actually increase muscle carnitine content. And they went on to show that this was associated with an increased expression of genes involved in lipid oxidation and iron CL turnover, as well as an increase in whole body fat oxidation during exercise relative to a control group. Now, the problem with this study, and you can kind of see it in this figure here, is that the control group here was actually just consuming carbohydrate beverage every day for 6 months. And the amount of carbohydrate required was very high. You can imagine feeding 2 daily beverages of 80 grams of maltodextrin. This control group put on weight, they put on fat mass, and in fact, you see a sort of blunting of fat oxidation. So what we're really showing in this figure is that taking carnitine with carbohydrate to increase muscle carnitine really blocks the detrimental effect of excessive carbohydrate loads in the diet. So that's an interesting finding, but it's not particularly useful or appropriate for targeting muscle carnitine in patient populations who are already suffering from obesity or insulin resistance. So during my PhD, one of my projects was primarily investigating whether you can still increase muscle carnitine with a lower carbohydrate load. And one of the challenges for this particular project was that you typically would feed carnitine for 12 to 24 weeks and take a biopsy and measure muscle carnitine content to see if it increased. And obviously this is a long time consuming process. So for this particular project, we tried to develop a more acute assessment of muscle carnitine retention. So we came up with this AV forearm balance model, so an arterial venous measurement of forearm carnitine uptake. So essentially, we're measuring the carnitine content in an artery, or in fact, in a vein that's been arterialized by heating, and then also in a deep vein draining from the forearm. So you locate this vein with ultrasound, and then you put a cannula in it. And this allows you to generate an arterial venous difference across the forearm, which is predominantly muscle. And combining this with a measurement of brachial artery flow using the ultrasound, you can get a handle on the actual carnitine balance across the forearm muscle. So it will tell you, is carnitine being taken up or released by the muscle. And we additionally combined this with the use of an isotopically labeled carnitine tracer, so a deuterated carnitine metabolite. And this will tell us about the absorption kinetics of carnitine. So the volunteers were just 8 healthy male students of healthy weight, young age. They came in, drank some water with the carnitine supplement, including the deuterated carnitine, and then an hour later consumed an insulinogenic beverage. So this was either just a controlled beverage with no calories in, or the carbohydrate beverage that had previously been used to increase muscle carnitine, or a beverage where we'd removed half of this carbohydrate and replaced it with protein. The idea being to try and still generate an insulin response, but with a lower glycemic load. So to go look at the results, first of all, I'm just presenting the data for the control versus the carbohydrate drink to sort of show a proof of concept. You can see, as expected, a strong insulin response when we feed carbohydrate. And then when we measure the forearm carnitine balance, you can see a stimulation of this positive carnitine balance in the carbohydrate group. So a positive number here relates to net uptake of carnitine from the muscle. So we see no stimulation of this in the control group, but a positive uptake in the carbohydrate group. So this was a nice sort of proof of concept that we can detect carnitine retention in the short term, and that this sort of validates the previous long-term feeding studies of carbohydrate to increase muscle carnitine content. Now, when we look at the protein trial, we can see that there was indeed a strong insulin response, but for some reason, this wasn't associated with any increase in the net carnitine uptake by the muscle. And in fact, we see it's sort of overlaid with the control group. So it appears that insulin response alone is not enough to drive the carnitine into the muscle. So we next looked at the plasma carnitine kinetics to sort of understand a bit more about what was going on. And you can see, both if we just look at the free carnitine concentration in the plasma following the drinks, or at the tracer enrichment in the carnitine, both were increased in the protein trial. Now, this graph doesn't tell us very much on its own because it's just showing the plasma carnitine. So we don't know if this is different because of increased absorption or reduced clearance. But because we have the tracer, the only way we can see an increase in carnitine enrichment is if there's an increase in absorbance. So this was a novel finding. It showed, for some reason, taking a protein drink with carbohydrate increased the bioavailability of the carnitine supplement, even though we didn't see any increase in the net carnitine balance. And then when we sort of combine these two measurements of the AV difference and the tracer enrichment, we get the following. So this is showing us basically what's the plasma carnitine enrichment in the arterialized blood or the venous blood. And there's no change in these measures in the control and carbohydrate trials, basically because the tracer is handled in exactly the same way as unlabeled carnitine. So whatever's going into the muscle is coming out of the muscle, is behaving in the same way. And that's kind of what we expected. Now, in the protein trial, you can actually see that the venous concentration of the tracer is lower than the arterialized venous concentration. And this is significantly higher than the carbohydrate or the control groups. Now, again, because we expect the tracer to be handled in exactly the same way as the cold carnitine, the only way you can see a dilution of the tracer in the venous blood is if there's unlabeled endogenous carnitine being shunted out of the muscle. So it looks as though what's happening here is, for some reason, when we give protein, you're increasing the carnitine efflux from the muscle bed. And you can kind of see that happening in the AV balance studies, but it's not really sensitive enough or noisy enough to detect it. Whereas with the tracer, you can see the significant reduction in the venous carnitine concentration of the tracer. So to summarize the findings from this first study, we say that oral protein appears to increase the bioavailability of the carnitine supplement, but may also blunt insulin-mediated carnitine retention. And this could be due to, for some reason, stimulating muscle carnitine efflux. So it may be that replacing carbohydrate with protein is not going to be sufficient to increase muscle carnitine in our patient populations. Now, the next study I'm going to talk about is one of these interventions where we increase muscle carnitine in a patient population. And this was in a cohort of older adults to see if we could increase lipid oxidation, IMCR turnover, and insulin sensitivity. And I will say that, unfortunately, when we started this project, we hadn't finished the previous project. So we did end up using a protein carbohydrate blend, which may have some impact on the results, as you'll see. So for the study design, we recruited healthy older individuals, so mean age of 69. They were overweight but not obese. And we conducted the study for 24 weeks, where they ingested L-carnitine daily in a eSure nutritional beverage. So that had a small amount of protein, carbohydrate, and fat in it. And we hoped that the insulin response was going to be sufficient to drive some carnitine retention. And this carnitine loading strategy was done concurrently with twice weekly supervised exercise sessions at a fairly low intensity. This was to make sure that both groups, whether carnitine or placebo, received some training benefit, and to try and increase lipid oxidation over the course of 24 weeks. And then at baseline and post-testing, we completed these metabolic studies where the volunteers were infused with a stably labeled palmitate tracer to monitor plasma fatty acid oxidation. They also had this combined with indirect calorimetry to measure whole body fatty acid oxidation. And then we measured the IMCL content in the muscle using electron microscopy, and we measured the insulin sensitivity with the gold standard measure, which is the euglycemic hyperinsulinemic clamp measurement. So first of all, just look at this graph on the left. This is the total muscle carnitine content. You can see that in the carnitine supplemented group, there was a significant increase of 20% in total muscle carnitine content, which was similar to what had been reported previously in healthy volunteers when this study was run. And when the resting RER was measured, which is a sort of measurement of gas exchange that sort of tells you whether you're burning carbohydrate or fat, there was a reduction in the resting RER in the carnitine fed group, which is indicative of a switch towards fat oxidation at rest. And consistent with this, when whole body fat oxidation was determined during exercise, you can see there's a 20% increase in fat oxidation. And this was consistent for all volunteers in the carnitine fed group. So this is whole body fat oxidation that was increased. We next looked at the tracer data to see whether this was coming from the plasma, because this 13C palmitate gives an indication of how much fat is utilized from the plasma and oxidized. And you can see there's no change either in the control or carnitine group. So what we're seeing here is that whilst whole body fat oxidation during exercise was increased by carnitine feeding, this wasn't due to an increase in plasma palmitate oxidation. So this tells us that this fat oxidation must have come from other sources, namely from intracellular lipids. So although it's an indirect measure, it suggests that IMCL turnover during exercise was increased by carnitine feeding. So did this impact on IMCL content in the muscle over 24 weeks? Unfortunately, it didn't. So measured by electron microscopy, you can see either the intramyofibrillar lipids or subsarcolemmal lipid fraction. Neither were appreciably reduced by lipid oxidation changes with carnitine feeding. And as you might expect, this translated into no difference in insulin sensitivity, either measured by the hyperinsulinemic clamp or by HOMA-IR. And there was no change in body weight or fat mass either. So to conclude this project, we showed that muscle carnitine can be increased by a dietary intervention in older adults. And this appeared to be effective in increasing whole body fat oxidation during exercise, but sadly, this did not translate into any metabolic benefits. And this could be because the increase in muscle carnitine wasn't high enough, or perhaps because there's not enough fat oxidation occurring over those 24 weeks. In other words, if they were doing more exercise sessions or at a higher intensity, perhaps the increased fat oxidation during those sessions would have a greater impact on IMCL content. So just to switch briefly now and talk a little bit about the work that I'm going to be doing with Professor Roach in the Conway Institute. I'm more interested in the impact of insulin resistance on liver metabolism in this study. And we're looking at the effect of metabolic inflammation on this progression of liver disease and whether this is mediated by the gut microbiome. So we know that certain dietary triggers, such as saturated fatty acids or sugars, can have a negative impact on the gut microbiome, whereas other triggers could have a positive effect. So we're looking at really whether the gut microbiome plays a role in driving the sort of metabolic and immune phenotypes in liver disease. And because my project is funded by the DevelopMed Precision Oncology funding, I'm going to try and link this to HCC, hepatocellular carcinoma. So very briefly, the first study we're conducting is comparing patients with just simple fatty liver to those with steatohepatitis, which is an inflammatory fibrotic condition. And by looking at their gut metagenome or their microbiome contents, we'll see if there's any link between this and their metabolic and inflammatory responses to dietary triggers, for example, high lipid tolerance tests or a high sugar tolerance test. So hopefully, if I do another one of these talks in the coming years, I might be able to share some of this data with you. And just before I finish, I'd like to obviously acknowledge most of the studies I presented today were done at the University of Nottingham, where my PhD mentors were Francis Stevens and Paul Greener. Francis is now at Exeter. And of course, the study participants that helped out. And these studies were funded by a BBSRC PhD studentship, and the aging study was funded by the Dunhill Medical Trust. And as I mentioned, Professor Helen Ross is my current mentor, and these studies are being conducted at UCD with the collaboration from Dr. Fred Sheedy at TCD and Professor Suzanne Norris, who's at St James's Hospital. So with that, I thank you all for listening and happily take any questions you might have.